everyone, I am again with you, myself Dr. Vivek Kumar, Professor, Himalayan School of Biosciences, Swami Rama Himalayan University, Jolly Grant, Dehradun. I am again with you with a new topic, Endophyte modulate host genome. So, we understand that the endophytes, they reside asymptomatically inside the plant tissues. Once they reside inside the plant tissues, it means they are going to influence the plant cell. The host plant, they develop intracellularly or intracellularly to complete part of their life cycle, and they have the endophytes, they have co-evolved with their host due to their crucial function in enhancing the health and survival of the plants. Endophytes benefit host plants by regulating photosynthesis, enhancing nutrient uptake and reducing the impact of various stressors. The various stressors, it could be biotic and abiotic, which we have discussed in the previous lectures. Now, first let us discuss the trans-kingdom signaling pathways. Trans-kingdom means endophytes, they are prokaryotes, host plant, eukaryote, different kingdom. Now, how they come in contact with each other, how they communicate with each other, how endophytes make an entry inside the host plant. The signaling communication between endophytes and plants, which helps creates a homeostatic phytobiome. It depends on communication between the two kingdoms. The endophyte recognition receptors are there, such as components of cell walls, extracellular ATP, apoplastic proteins. All these could generate synergistic signals with their host plants during the interaction between transkingdom. So, in addition, endophytes, they also evade plant immunological signals and colonize tissues to form symbiotic relationship with the plants and can prevent the plant pathogenesis. Consequently, the endophytic symbiotic signals and their tolerance to immune alteration of immune host and resisting pathogens qualify them to acclimatize and uphold homeostasis in the host plant. So, they are valuable endophytes, they can escape pattern recognition receptor PRR, detection by developing different microbe assisted molecular pattern MAMPs and altering cell wall polysaccharides and chitin constituents, thus repressing plant immunity to endophytic microorganisms. Additionally, endophytes, they can disrupt host immunological signaling by secreting effectors, including the apoplastic protein effector and extracellular ATPs. So, the host plant sensing of microbial cell wall components is the primary factor in recognizing oomycetes and fungal pathogens. Arbuscular mycorrhizal fungi, AIM fungi, they produce short chain chitin oligomers and lipocheto-oligosaccharides, signaling derived from microbial cell wall and exudates, which can be detected by lysine motif (LysM) receptors intricated in activating common symbiosis signaling pathways. The exopolysaccharides, lipopolysaccharides, capsular polysaccharides, cyclic glucons and other microsurface polysaccharides, they are significant indicators of plant microbial interactions. Chemotaxis. Initially, the methyl accepting chemotaxis proteins MCPs, they play a vital part in the chemotaxis of free living bacteria toward the host root. The Herbaspirarum, the Chi SMR1 Chi inactivation mutant, it demonstrated the role of MCPs in plant colonization. So, out of 66 MCP encoding genes, 9 genes in SMR1 expressed in root associated cells. So, attachment of mutant strains, it was reduced by 2 times when one of these HSERO 3720 was inactivated compared to the wild type strain. This MCP plays a chief or main transducer needed to recognize the rhizospheric zone and guide bacteria toward the host excreted components. Conversely, the SMR1 mutant endophytic and epiphytic strains, colonization was identical to that of the wild type. The numerous type of bacteria, they are drawn to root exudates and curly proteins impact bacterial cell adherence to the surfaces, cell aggregation and biofilm development. So, the initial stages of attachment and colonization, the bacterial membrane, proteins and lipopolysaccharides, they play an essential part. The monosaccharide rhamnose typically found in LPS and O antigen it requires 4 gene RFB ABCD for production, and in early colonization stage LPS biosynthesis genes they are upregulated. Okay. Mutation in gene, if we imagine that there is a mutation in the gene, then mutation in one of the genes implicated in the rhamnose biosynthesis. Rhamnose is sugar. RFB or RBC leads to lesser endophytic colonization of maize by Herbicillarum SMR1 and about 100-fold less attachment to root surface. Oxalate is found in maize and lupin plant root exudates, and with the help of oxalate decarboxylase, this enzyme is produced by the endophytes, and using the enzyme the bacteria could utilize the oxalate. So, early colonization of lupin and maize was reduced when Burkholderia phytoferments OXC genes which encodes oxalate decarboxylase, it was deactivated. However, the effect was less noticeable in the maize and it was more pronounced in the lupin plant. 2A, 2B genes were activated in Rhizobia meliloti on the infection threat to the root surface. The disruption of one of these genes reduced the root's capacity for colonization, showing the importance of tree halos and or multi-toll utilization. So, during host colonization by different microbes, the host may also stimulate the diverse clusters of gene expression during the process of colonization. Now, endophytic Enterobacter species is described as possessing transporting iron, amino acids, hemagglutinin, hemolysin genes which are required for endophytic and plant communications. Endophytic bacterium Bacillus aryabhatti, which brought transcript changes in Arabidopsis and promoted Arabidopsis and tobacco plant growth. So, several plant genes including berberine bridge enzyme like gibberellin regulated protein, cinnamyl alcohol dehydrogenase, benzaldehyde dehydrogenase, epirase and other enzymes, they were substantially expressed after the priming with the endophytes. Now, plant growth and development. The plant growth promoting endophytes, they occupy diverse plant tissues and they establish a beneficial association with the that expedites the exchange of nutrients and progresses and they progress the other accomplishment within the host plant. So, one of the well studied methods of promoting plant development is the endophyte synthesis of phytohormones. These phytohormones they alters the morphology and structure or architecture of the plant host. So, endophytic microbes they produce nitrogen, they solubilize insoluble phosphate, potassium and they compete with phytopathogens for same environmental niche. Endophytic bacteria Stenothermoplasma, Stenotrophomonas, Rhizophila isolated from Glycyrrhizia seeds acted as a potential biocontrol agent against pathogenic Fusarium acuminatum. Inoculation of Stenotrophomonas repress Fusarium wilt and encourage the host plant growth. And then the transcriptomic analysis discloses photosynthetic molecular regulation in Acantharum by epichloi endophyte under Bloomeria infection. So, in this Bloomeria infection, epichloi presents upregulated genes which are involved in the photosynthetic activity of the Acantharum in a Rien's plant. Medicinal plant Pulicaria usually inhabit the found in the arid regions from which several bacterial endophytes have been isolated like Agrobacterium, Acinetobacter etc., etc. These endophytes were shown to produce different secondary metabolites, indoleacetic acid, ammonia, extracellular hydrolytic enzymes and they also solubilize the phosphate. And these endophytes they also exhibited in vitro antagonistic activities against 3 phytopathogenic fungi, Fusarium, Alternaria and Pythium species. Now, if this picture shows the endophytic effect on the host plant, the endophytes they fix nitrogen, they solubilize phosphate, they transport mobilize the movement of iron and zinc, produce secondary metabolites and they control the growth, modulate the growth of the plant by producing the phytohormones. Endophytic fungi, several endophytic fungi like Epococcum, Sporomelia etc., they enhance Pova pratensis and Trifolium plant growth. And it was observed that endophytic fungi enhance pasturage yield and nutrient contents like these metals cadmium, lead, zinc, copper, manganese, calcium etc., and total S content of forage compared to the control. Now, the plant abiotic and biotic stress tolerant mechanisms, these all are modulated by the host plants endophytes. The inoculating plants with endophytes that can produce ACC deaminase reduces the level of ethylene, protecting the host plants from saltiness and premature senescence. Crop productivity and yield have been increased under saline environments by using salt tolerant or salt resistant endophytic microorganisms. So, in one experiment, influence of salt tolerant Bacillus abylofaciens SN13 strain on rice plant was observed in hydroponic and soil environment under saline conditions, NaCl up to 200 millimolar. This strain SN13 augmented plant growth, mitigated salt stress and modulated differential transcription of at least 14 genes. Out of 14 genes, 4 genes were upregulated and 2 they were suppressed in hydroponic salinity conditions. The osmotic control during salinity stress is aided by the accumulation of osmolytes and polyamines. Now, salinity mitigation in Triticum stivum, it was observed when inoculated with endophytic fungi Pyremospora indica, endophytic bacteria Micrococcus luteus, when manufactured synthesized indolacetic acid, Bacillus methylotropicus synthesized a greater amount of exopolysaccharides, Bacillus equimaris demonstrated appropriate ACCD production. So, different types of microorganisms, they produce different types of secondary metabolites. The endophytic bacteria increase the root biomass and lateral roots of Helianthus and produce the secondary metabolites ACCD, IAA, exopolysaccharide under water scarcity conditions. Now, the endophyte they also induce plant defense genes. The endophytes offer the prospect of an active microbial contribution as it leads to the host genome modulation, ensuing induction of numerous modifications in the host cell wall, that is build up of cellulose, callose, pectin and phelonic components. All these events result in structural barrier formation at the location of a possible strike by the pathogens. So, once these barriers are formed, then obviously the pathogens they are unable to make their entry inside the host plant. Likewise, the endophytes produce cell wall degrading products or defense associated enzymes such as chitinase, peroxidase, beta-1,3 glucanase. This picture shows the cell wall degrading enzymes of endophytes, and these cell wall degrading enzymes, once they are produced, then they affect adversely the pathogens. The endophytic microbes they can affect how plant express their genes, causing physiological changes that in some situations boost a plant's resistance to stressors which is brought about by the climate change. For instance, ecological stresses can enhance plants' reactive oxygen ROS production. Microbes can alter gene expression which are integrated in ethylene biosynthesis and ROS scavenging, boosting photosynthesis of the host plant and plant growth to ameliorate stress such as drought, heavy metals and salinity. Endophytic fungi Metarhizium, it has been reported to regulate defense related genes expression and the phytohormones contents in maize plant which have been primed with pathogenic fungus Koch leobolus. It was proposed that Metarhizium promoted maize growth and reduced southern corn leaf blight development. Probably by inducing the endophytes, this Metarhizium has been probably carried out this function by inducing systemic resistance which have been facilitated by phytohormones regulation and expression of defense and growth associated genes in the maize. Finally, the keywords. The endophytes are a great community that involves a diversity of species, each with distinctive morphological, physiological functions and therefore variable symbiotic associations with their corresponding host. And it has been generally consented that most endophytes help in growth endorsement of the host plant, and in their particular host they play particular functions. So, this endophytic competency with their host intimates their ability for diverse functions such as nutrient mobilization, phytohormone production, they act as biocontrol agent. Besides them, they perform other beneficial functions in the host plant which enhances the host plant growth by mitigating the abiotic and diverse biotic stresses. Thank you so much.